Dear children, welcome to biology online classes. In the previous class, we have learned about various kinds of biomolecules. I told there are different biomolecules which make up the living organism. So, living organisms are made up of cells we have studied, isn't it? And we were studying about the various components of cell. And when we analyze the chemical nature of these components, like plasma membrane or what mitochondria, so all this contains what certain molecules, and these molecules which make up the living organisms, okay, is known as what biomolecules, okay, and they are mainly carbon compounds and that we get from living tissues are can be called as what biomolecules. So in addition to this, we have inorganic compounds also. We have discussed all this, isn't it? Minerals are there, isn't it? That can be obtained by what? By burning the living tissues, isn't it? So inorganic elements like calcium, magnesium, etc. are also there. And we have studied during the chemical analysis, we get different kinds of molecules, and one among was what? Amino acids. Okay, so amino acids are what? Building blocks of proteins are made up of amino acids. Like a chain is made up of different beads, isn't it? The polypeptide chains are formed by the union of what? Amino acids. So when we study about amino acids particularly, amino acids are what? Substituted methane. Methane's formula, CH4, isn't it? The four valencies of carbon is filled by hydrogen atoms. But here in amino acids, this carbon valencies are substituted by different what groups, like what hydrogen, carboxyl, amino groups, okay, so and a variable group known as R group. So these what various valencies of carbon is substituted by this kind of groups or compounds. Okay, so based on that, I have told you there are acidic amino acids, isn't it? Then there are the number of this amino groups or the acidic group. Okay, if the acidic group or COH group is more, then that amino acid will be acidic, and example we have studied. And if it is basic, means if amino group is more, then it is basic, and if the number are same, then it is what? Neutral amino acids. Clear? And there are aromatic amino acids when there is what? Aromatic compounds substituted in their valencies. Understood? So another important feature I have explained you, that is the ionizable nature of NH2 group and COH group. And due to this, in solutions of different pH, the structure of amino acids varies, and it can exist as Switter ion. So what is Switter ion? Actually, Switter ion is amino acid in which both the charges, positive and negative charges, are there. Okay, so the H ions, hydrogen ions from COH group will be shifted to this, and it will get a what? Negative charge, and H plus ions, as it is getting added here, this will have a positive charge. So this is called what? Switter ion. So about amino acids, you understood now clearly. So amino acids are building blocks of proteins, I told. So we'll directly study the proteins. Okay, so there are what? Lipids, okay, nucleotides, etc. So that we will study later. Let me teach you about the proteins first. So we'll go to that portion of this chapter. So directly I'm teaching you the 9.4, okay, proteins, okay. So that will give you some what? Continuation. That's what I'm teaching you. You please take this page and read. Okay. Proteins are what? Polypeptides. Understood? Polypeptides means what? Polypeptides are formed by the joining of amino acids. And this amino acids in a polypeptide chain, okay, is linked by peptide bond. Understood? So peptide bond is That is CONH bond is present in between two adjacent amino acid in a polypeptide chain. Understood? So, COH is having OH. Okay, the OH of COH and one H of NH2 group. They will what combine and they will be getting eliminated as water. Okay, and what will form? CONH bond. This bond is known as what? The peptide bond. And I told you protein is a polymer of amino acids. And there are 20 amino acids. Okay, 20 types of amino acids. So some examples we have learned, isn't it? Alanine, glycine, cysteine, proline, tryptophan, lysine. All these are what? Different types of amino acids, and there are 20 amino acids. Okay, and because of the presence of different amino acids, protein is always a heteropolymer. Polymer means what? Which is made up of or which is formed by the union of many units called monomers. Understood? So always what protein is a heteropolymer, and it's not a homopolymer. Okay, homopolymer means what? If it is having a same type of monomers repeating many times, then it is known as what? Homopolymer. Heteropolymer means if it is made up of different types of monomers. So as there are 20 amino acids and they are found repeatedly in a polypeptide chain, the proteins are examples of homopolymer. Polymer means what? A molecule which is formed by the repetition of what monomers. Okay, so if the monomers are of same type, then we call it as what? Homopolymer, and if it is of different types, we call it as heteropolymer. Is it clear? Then, so in our body, okay, some of the amino acids are essential and that should be supplied through our diet. So, the dietary proteins means the proteins which we eat, okay, they are the source of essential amino acids. So, the amino acids can be essential or non-essential. Okay, so essential means what? Which are needed, isn't it? Which are needed to get means which are needed by our body, and it should be provided by the dietary proteins, means the proteins which we eat. From that only we will get that. And non-essential are those which our body can make. Understood? So the essential amino acids we cannot make, our body cannot make, and non-essential ones are what in they can be produced in our body, or our body can produce. So what are the functions of proteins in our body? Okay, some we learned in plasma membrane there are proteins, isn't it? Integral proteins and peripheral proteins, and they are acting as what? They are acting in the transport of materials across the cell membrane. Some fight infectious organisms, isn't it? Antibodies you would have heard. And some are hormones. Hormones are there which are proteinaceous in nature. So, and most of the enzymes are proteins. Clear? So likewise, what a number of functions they do, means structural functions. Then they do the functions of, or they do different physiological functions. Likewise, number of functions they do in our body. Okay, so collagen is the most abundant protein in the animal kingdom. Okay, and ribulose biphosphate. Okay, carboxylase or what oxygenase. Okay, ribulose RUBP. We will study this also in the respiration chapter. Okay, ribulose biphosphate carboxylase oxygenase, rubisco, okay, is the most abundant protein in what? In the plant kingdom or in all of the means biosphere. Okay, in animal kingdom what is it? Collagen. Okay, and in the whole biosphere, which is that? RUBP. Okay, RUBP carboxylase or oxygenase. RUBP carboxylase or oxygenase, that is, in short we call them as rubisco. Is it clear? So about this you understood now. Now I will teach you 9.7. Okay, that is structure of proteins. Okay, so please take this page. So proteins are heteropolymers. I told what are heteropolymers, because they are made up of different types of monomers, isn't it? And the structure of proteins, if you see by a biologist, okay, so what there are what the molecular formulas, isn't it? Then we can represent the structure of a molecule while we are saying that in organic chemistry, or in three-dimensional views it can be understood. So according to a biologist, okay, the structure of proteins can be explained in four different levels. Okay, so this can be asked, describe the structure of proteins. So the various structures that explains the proteins, okay, or the different ways by which the protein structure is explained are the primary structure, secondary structure, tertiary structure, and the quaternary structure. Understood? So let's see what all these are. So the first is what? The primary structure. So primary structure of a protein means what? It describes the number, okay, and the sequence of amino acids. So I told, in a polypeptide chain the amino acids are arranged, okay, or what, one by one. Isn't it? Amino acid 1, 2, 3, likewise it is arranged, isn't it? And when it is arranged like this, the left end, that is represented by the first amino acid, and the right end, the last end of the, means the right end, okay, the last right end is represented by the last amino acid. And the first amino acid is called the N terminal, and the last amino acid is called the C terminal amino acid. Okay, so in this, how many amino acids are there? Understood? So you can see here in this, okay, the first amino acid is what? And second, third, fourth, likewise, they are arranged linearly in a polypeptide chain. So the number, how many number are there, and how, what is the sequence? Means first, for example, alanine, if it is there, then next what comes? Okay, so their sequence, which comes first and which is in the next, likewise the number and sequence of the amino acids in a polypeptide chain is explained by the primary structure. Is it clear? Now the secondary structure means, so a protein thread or a polypeptide chain is not arranged as a rigid rod. Understood? So it will be folded in its own axis, okay, in different ways, in the form of helix, or in case of sheets, pleated sheets, beta plated sheets. Okay, like in different ways, this chain itself can be arranged. So this is explained by the secondary structure. Understood? So which forms, in which manner the polypeptide chain, which is linear in nature, is arranged in the two-dimensional way. Okay, in the two-dimensional structure, which explains the foldings of a single polypeptide chain, is known as what? The secondary structure. So this is the primary structure of a protein. You can see they are having what? Different amino acids, okay, in N terminal and in the C terminal. Okay, you can see different amino acids are arranged. Okay, now a secondary structure means what? Now in between what this what amino acids, okay, hydrogen bonds can be formed, and they can get folded. Okay, they can get folded into alpha helix or in the beta pleated structure. The same polypeptide chain will get folded, understood, to form helical structure or alpha or beta pleated structure. Understood? So this is known as the secondary structure. Okay, so this is the what? Secondary structure. Understood? The polypeptide chain in its own axis, okay, it will what? Fold into the alpha helix or in the beta pleated structure. Is it clear? This is what the tertiary structure of proteins. Okay, so primary structure you know, secondary structure you know, and the arrangement of proteins in the 3D space is called what? The tertiary structure. You can see here it is getting folded in 3D and now it is what? Tertiary model, okay, or tertiary structure. Now some proteins are having more than 2 polypeptide chains. Okay, if a polypeptide chain is folded upon itself like a hollow woolen ball, okay, then it is known as what? Tertiary structure. Okay, so here also you can see it's the primary structure, okay, which explains the number and the sequence of amino acid. Okay, how many amino acids are there in which sequence they are arranged. Then secondary structure explains what? The folding of this particular polypeptide chain in its own axis. And tertiary means what? In space or in the 3D form, how it is getting folded. Okay, now next is what? The quaternary structure. So quaternary structure explains what? No, the manner in which there are, means if there are more than one polypeptide chain, and in which manner these individual polypeptides are folded. Okay, so that gives you the quaternary structure. Is it clear? So all this, the example for this is human hemoglobin. Okay, hemoglobin is having actually 4 subunits. Okay, 2 of this are identical and 2 are another 2, they are also identical. So there are 2 alpha polypeptide chains and 2 beta polypeptide chains. Okay, so all together how they are arranged, that express or that explains the quaternary structure. So what I explained today is the structure of proteins. Okay, so what are the different structure of proteins? Primary structure explains what? The number and the sequence of amino acid. Okay, and I told the left end is representing the first amino acid and the right end express the or indicates the last amino acid. The first amino acid is known as N-terminal and the last amino acid is known as the C-terminal amino acid. Okay, so a protein thread, it can get folded in the form of a helix. Okay, and only some portion of the protein threads are arranged in the form of a helix. And in proteins, right-handed helices are found. Understood? And other regions, they are folded into other forms, and that gives what? The secondary structure. Means the polypeptide chain, they fold in its own axis in different ways, either in the helical manner or in the pleated manner. Understood? So in that way it will give what? The secondary structure. Then the long protein chains can also get folded upon itself like a hollow woolen ball. You would have seen the woolen ball, isn't it, in which the woolen thread is arranged. So in that way it can get arranged it's by its own. So that is representing the tertiary structure. Then quaternary structure is explaining, okay, or this kind of structure is there for those proteins which are having more than one polypeptide subunits. Understood? So the manner in which these individual folded polypeptides are arranged with respect to each other, how they are arranged. Okay, this polypeptide chains, how they are arranged, that gives what? The quaternary structure. So about the different structure of proteins, I hope you clearly understood. Okay, so we'll continue in the next class. Thank you very much.